team is back with another episode of Topical Explainers for the second chapter of CAIE, A2 level biology. In this video, we'll be discussing all about photosynthesis, simplify the topic, and provide us with all the information we need. We have Zhen here with us.
Hi guys. So yeah, as you said, we will be doing photosynthesis today, which is quite a short topic actually. It's quite a lot shorter than respiration. It's probably the shortest topic of the entire A2 biology syllabus. So yeah, today we'll be talking about the Calvin cycle, photophosphorylation, and the limiting factors, and some of the limiting factors of that can affect photosynthesis in plants. So yeah, anyway, let's get on to it. So what do you know about photosynthesis?
So one thing we know is that it occurs in the chloroplast, and chloroplast contains chlorophyll, and the overall equation of the reaction is 6 CO2 plus 6 H2O, which is carbon dioxide and water. Within presence of light energy, and of chlorophyll, it would give us C6H12O6, which is carbohydrate or glucose, plus 6CO2, which is oxygen.
Yeah, exactly. It's a relatively simple equation that we have learned in the past in IGCSE, or even earlier actually. But do be aware, we'll go much deeper into this today. And this is just a summary of the equation. So yeah. So let's take an outlook of photosynthesis. So as I said, the reaction shown in the overall equation is actually made up of many other smaller steps. These can be divided into two main stages, the light-dependent stage and the light-independent stage. So as you can see here, this is your light-dependent stage. So light is used in this stage, and here's your light-independent stage. See? And yeah, here's your overall equation. So what happens in the light-dependent stage is that light is absorbed by, as you know, these things called chlorophyll, and yeah, they generate ATP and NADPH, which is fed to the light-independent stage, which includes the Calvin cycle. So we'll go more in depth into this. So first of all, we should take a real look at the properties of our chloroplast. So what is there? What is in there actually? So yeah, so in the chloroplast, here, the fluid within the chloroplast, we call it stroma, and it contains the enzymes that are required for the reactions of the light-independent stage of photosynthesis, which, for example, is rubisco, which you'll learn later on. And there are also small ribosomes and small circles of DNA, which is used to synthesize proteins. And there are also starch grains, which store some of the carbohydrate made in soluble form. As you know, glucose is soluble, and so is sucrose, but starch is actually insoluble. So it's a good storage, it's a good form of storage for energy. And yeah, and if you notice, the image of the chloroplast actually kind of resembles the mitochondria. So in this case, we can somewhat refer to the stroma as kind of the matrix, as equivalent to the matrix of in the mitochondria. So yeah, stroma, chloroplast, and your matrix, mitochondria. So yeah, the membranes of the lamellae and thylakoids hold carrier molecules that work as an electron transport chain for the photophosphorylation / chemiosmosis. Now, where have we seen chemiosmosis before?
We've also seen it in mitochondria, right, from the previous chapter.
Exactly. So it's the exact same kind of process, like chemiosmosis, to generate ATP. And yeah, but in this case, instead of oxidative phosphorylation, we call it photophosphorylation. Why do we call it photophosphorylation? Well, what does photo mean? Light, right? So yeah, we call it kind of like light phosphorylation. So light is used for this entire process. And yeah, anyway, so the lamellae also have different photosynthetic pigments embedded within them that absorb energy from certain wavelengths of light. Mm. So yeah, let's take a little look at how the lamellae and the thylakoids actually look like. So as you can see here, labeled is your thylakoid and your lamella. The lamella is essentially the inner membrane, not inner membrane, but like it's kind of like a membrane structure inside your chloroplast, and your thylakoids actually stack together to form something called your grana. Yeah, it's here. Your grana are stacks of thylakoids, as I said, that hold photosynthetic pigments. So what is the purpose of these grana? Well, like the cristae that we find in your mitochondria, they actually have a large surface area. Why? In this case, it's actually to maximize light absorption. Yeah. And different photosystems absorb light at different wavelengths. So essentially, why is there why can we maximize light absorption with a larger surface area? Is because there are more of these kind of photosystems. What is photosystems? Well, we take a look on the later slides. Anyway, a few things to know. A few things to know before we get into all of the more complex type stuff. So yeah, energy is transferred as ATP and reduced NADP from the light dependent stage and is used during the light independent stage, which includes your Calvin cycle, of photosynthesis to produce complex organic molecules. So yeah, essentially what this statement is talking about is that when uh, light is absorbed by by all the pigments in the thylakoids, they generate ATP and your reduced NADP. What is reduced NADP actually? Well, it's actually similar to what we've learned in chapter 1 of A2 biology. You've learned that in mitochondria, there are these things called hydrogen carrier molecules called NAD and FAD, right? Well, in this situation, it's actually it acts as the same thing. In this case, we call it NADP in plants. And yeah, these two things are passed on to your Calvin cycle, and to produce complex organic molecules, and of which we will take a look on on the later slides. Anyway, within the chloroplast, the thylakoid membranes and thylakoid spaces, which occur in stacks called grana, are the site of light-dependent stage. See, as you can see here, your light reactions occur in thylakoids. See, this is actually a kind of small image, not really a small image, but like a simple image of how your thylakoids actually look like. See, you have individual plates, kind of discs, and all of these stacked together is called your grana. And within the chloroplast, the stroma is the site of the light independent stage. Yeah, inside all of this, the fluid inside the chloroplast. So I've mentioned about the word pigment before, right? So what are they? So we'll take a look on accessory pigments. What are accessory pigments actually? Well, here are some of the examples. They're also called antenna pigments, you can see. They are xanthophyll, carotenoid, and chlorophyll B. So these pigments in thylakoid membranes are arranged in clusters, or I said, as previously said, photosystems. So what this sentence sounds kind of complex, but it's actually not that complex. It's just what we call photosystem. It's just a group of these pigments, light absorbing pigments, and they are arranged in clusters. So essentially groups of these pigments. And there are two types of photosystems: photosystem 1 and photosystem 2. Photosystem 1 mainly absorbs light of wavelength 700 nanometer, and photosystem 2 mainly absorbs light of wavelength 680 nanometers. This is actually quite a specific point that you may have to remember, because it has come out in the past exams in the mark schemes. So yeah, it's good to remember these values. So as I said, these accessory pigments, right, they absorb energy from light and channel it to a reaction center. Quite an interesting, uh, fact here, right? So what they essentially do, to kind of simplify it, is these groups of pigments, accessory pigments, they absorb the light. As we see, right, in photosynthesis, chlorophyll absorbs light. Well, actually, there is actually these pigments. There's so many of them. Your xanthophyll, your carotenoid, and chlorophyll B. They absorb these light, and they all transfer it as energy, right? Light energy. They convert it and transfer the energy into your reaction center, this green circle here, of which you can see in simplified format is called contains chlorophyll A. Yeah. So one reaction center contains two molecules of chlorophyll A. So actually, if you see here, right, you can see here, you have accessory pigments, which is chlorophyll B, and uh, and chlorophyll A. These are actually very different things. One is an accessory pigment, and uh, one is in the reaction center. So what is the difference actually? Well, chlorophyll B, chlorophyll B, they chlorophyll B is an accessory pigment. It absorbs light, whereas chlorophyll A actually receives the energy from that is receives the energy that was absorbed by the chlorophyll B. So the chlorophyll B transfers the energy, the light energy, to chlorophyll A. Why is that? We'll take a look on later. So let's take a break from all that complex stuff and take a look at something slightly more simple. Your absorption spectra. What is that actually? Well, it's just a graph showing the absorbance of different wavelengths of light by a photosynthetic pigment. See, you can see chlorophyll A, chlorophyll B, carotenoids. Quite interesting. But yeah, there's not much to it. You just have to. It's just essentially what it is. It's what it has been said, and the definition lah. Your graph showing the different the absorbance of different wavelengths of light by a photosynthetic pigment. And yeah, then the second graph that you have to learn about in A2 biology, photosynthesis, is your action spectra. Previously, you have your absorption spectra, right? The absorption of light. Now you have your action spectra. So what is it actually? Well, it's just a graph showing the effect of different wavelengths of light on a process. For example, the rate of photosynthesis. Yeah, and it shows the rate of photosynthesis at different wavelengths of light. So you can see it correlates with the absorption spectra. The more the absorbance, as you can see here, the more the rate of photosynthesis, or yeah, essentially that. You can see here, the peaks are at the same exact position. So yeah, it's quite a simple type of outlook here. And yeah, so let's talk about the more juicy parts about photosynthesis now. The more things that might actually come up. So yeah, your light dependent stage. As I said previously, there are two parts, there are two stages when it comes to photosynthesis, two main stages per se. So yeah, your light dependent and your light independent. So we'll begin talking about light dependent stage first. So cyclic photophosphorylation and non-cyclic photophosphorylation occur during the light dependent stage of photosynthesis. And yeah, these 4 points here are kind of like a summary for the light dependent stage. Kind of like an intro to the more detailed parts later on. So yeah, the first point, splitting of water by photolysis to give hydrogen ions, or in other words, protons, by oxygen evolving complex, or you can say water splitting complex. Quite an interesting word, right? Quite an interesting phrase, of which we'll learn later on, more detail. Which, as here says, which is found in photosystem 2, so only occur at non-cyclic photophosphorylation. And then your second point is synthesis of ATP in photophosphorylation, hydrogen ions combine with NADP to make reduced NADP, and your ATP and reduced NADP are passed from the light dependent stage to the light independent stage. So yeah, that's kind of a sum up of your light dependent stage for now, and we'll move on. Photophosphorylation and chemiosmosis. As I said previously, photophosphorylation, it's a similar process to oxidative phosphorylation. So what are the main points again? Well, it's kind of like a repeat. So it's actually a good revision for it's quite a good revision for your chapter 12 as well, respiration, because in this case, in respiration we talked about oxidative phosphorylation, and now we'll be talking about photophosphorylation. So yeah, energetic electrons release energy as they pass through the electron transport chain. Sounds familiar, doesn't it? The released energy is used to transfer protons across the thylakoid membrane in creating a proton gradient, very similar to oxidative phosphorylation, except that instead of the protons are transferred across the inner membrane of the mitochondria, it's now through the thylakoid membrane in your chloroplast. And protons return to the stroma from the thylakoid space by facilitated diffusion through ATP synthase, providing energy for ATP synthesis. Yeah, very, very, very similar to oxidative phosphorylation. And let's recite the stages for chemiosmosis again. So excited electrons are captured by an electron acceptor in the thylakoid membrane and passed along a chain of electron carriers, which are your electron transport chain. The carriers are alternatively reduced, electron gain, and oxidized, electron passed to the next carrier. Electrons gradually lose energy as they pass along the chain. Then energy used to actively move protons from the stroma across the thylakoid membrane in the thylakoid space. Protons move down their concentration gradient by facilitated diffusion through ATP synthase molecules. ATP is synthesized by adding phosphate ion to ADP. To be honest, this entire process is just a more expanded, more detailed version of this process right here, because essentially you can view them as somewhat the similar process, but Yeah. So let's move on to your very main, like very main topics of today. Cyclic photophosphorylation. Hmm. So what occurs here? What is it actually? Well, you can see it's cyclic. So it occurs in a cycle. Oh, like that. Occurs in a cycle. And photophosphorylation. So in cyclic photophosphorylation, only photosystem 1 is involved. Remember that. These points are very important, and you can memorize it and try and understand it when I explain it. So what happens in cyclic photophosphorylation? Well, photoactivation of chlorophyll occurs. What is photoactivation? So let's split the word into two parts. Photo, light. So light is used. You know that light is used. Anything with photo in it means light is used. Activation. What words are synonyms of activation? Active. Excite, right? You excite something. So photoactivation actually is just, in this case, uh, to activate the electron in, as I said previously, in the reaction center, or more specifically, your chlorophyll A of your photosystem. Remember the clusters of photosynthetic pigments, so that it can be emitted. Light energy is absorbed by photosystem 1 and is passed to the reaction center, as I've mentioned. And energy absorbed excites an electron in the chlorophyll A molecule to a higher energy level. So what happens? You might actually learn this in chemistry, but what occurs is that your when energy is absorbed by your by the electron in the chlorophyll A molecule, so your electron has more energy, right? It has reached a higher energy level. And since this electron is now higher in energy, the electron is emitted from the chlorophyll. It has so much energy that is excited. We call it excited. Remember that word. Excited. And then since it's excited, it is emitted from the chlorophyll A molecule. Chlorophyll A molecule. So this is called, as I said, photoactivation. So in this entire process, you can see here, when electron is emitted from your photosystem 1, it passes through your is uh, accepted by your first electron carrier and passes through what did we learn? Your electron transport chain. And in that entire process, your ATP will be synthesized by photophosphorylation, or in other words, chemiosmosis, as was mentioned in the previous slide. But in this case, no reduced NADP is synthesized, which is exactly the very point of which separates your cyclic photophosphorylation and your next slide, non-cyclic photophosphorylation.
By the way, in the
So
previous slide, does photolysis not occur?
yes, photolysis actually does not occur in cyclic photophosphorylation, but does occur in your non-cyclic photophosphorylation. Why is that? Well, actually, the entire purpose of photolysis is to split photolysis, right? Is to split water into oxygen, hydrogen ion, and an electron. This electron is used to replace the missing electron of chlorophyll A when the old electron, we can say it's old electron, has been emitted from the photosystem, from the chlorophyll A. But in this case, in cyclic photophosphorylation, you can see that your electron is actually replaced by itself. It's a cyclic photophosphorylation. See, your electron, after it's emitted and passed through the your electron transport chain, actually returns to your reaction center. But that is not the case in non-cyclic photophosphorylation. So what occurs in non-cyclic photophosphorylation? Well, the first distinguishing factor between cyclic photophosphorylation and non-cyclic photophosphorylation is that it not just only includes photosystem 1, but also includes photosystem 2. So photosystem 1 and photosystem 2 are both involved, the two types of photosystem that we learn. So as I said, it's kind of similar. It's a very similar process to previously, except that it's non-cyclic. Is the process does not form a cycle. See, so we actually call it the Z structure in quite an interesting name. But yeah, photoactivation occurs in the reaction center of photosystem 2, resulting in your electron being emitted. So the first photosystem that is involved is actually your photosystem 2. Your electron is photoactivated and then it's emitted and is actually accepted by your first electron acceptor, and yeah, photophosphorylation occurs, chemiosmosis occurs, and your ATP is generated. Hence, as it says here, ATP formed from ADP and phosphate ion. So yeah, electron is accepted by first electron acceptor and passes through your electron transport chain, generating ATP in the process of photophosphorylation, or in other words, chemiosmosis. So the electron of photosystem 1 is also emitted via photoactivation. So same thing. And so the electron from photosystem 2 replaces it after passing through the electron transport chain. See, your electron here replaces the electron in your reaction center after it has been emitted and passed through your electron transport chain. And the electron emitted from photosystem 1 passes through another series of electron transport chain, also contributing to the photophosphorylation. And your electron is then combined, well, after it passed through the chain of electron carriers, is then combined with your NADP and proton. Is combined with your NADP and your H+. Where do we get the H+ from? Well, it's from your photolysis of H2O. See, you still have your extra H+ here to form reduced NADP. So when photolysis occurs, as I said before, your water splits into your oxygen, hydrogen ion, and electron. So yeah, kind of the basis, everything you need to know about cyclic and non-cyclic photophosphorylation. And there's actually a bit more here. ATP and reduced NADP are synthesized, energy emitted from photosynthesis. These are just the properties of non-cyclic photophosphorylation. So yeah, ATP and reduced NADP are synthesized. Energy emitted from photosystem 2 is used to form ATP. Energy emitted from photosystem 1 is passed to the coenzyme NADP, producing reduced NADP, as aforementioned. And your electron lost from the photosystem must be replaced. You cannot leave it hanging, as I said. But yeah, the missing electrons in your reaction center must be replaced. So, and photosystem 1 receives the electron that was emitted from photosystem 2, and photosystem 2 receives the electron from the photolysis of water. I've never said this point before. So yeah, so when photolysis occurs, your water splits, right? Your H2O splits into oxygen and your H plus, and your electron fills the missing spot of the reaction center in photosystem 2 after the electron has been emitted, after the old electron has been emitted from your chlorophyll A molecule. So what enzyme actually catalyzes the photolysis of water? Well, we call it the oxygen evolving complex or water splitting complex. Is actually an easier name, but I do prefer the oxygen evolving complex, as oxygen is evolved from this process. O2 is a waste product. See, O2 is released. Hence, in your in previously mentioned the summary, the overall equation, your 6O2, comes from here. The thing that we breathe. So we've talked a lot about light dependent stage. Now let's move on to your light independent stage, which includes your Kelvin cycle. So light independent stage of photosynthesis. It does not itself require energy from light, but requires inputs from the light dependent stage. ATP and reduced NADP are used to convert CO2 to carbohydrates. So what is the entire purpose of Kelvin cycle? It's actually to produce, as I said previously, to produce organic molecules, such as your glucose, your amino acids, and your lipids. So it's kind of like a kind of generator of these kind of molecules, that is used by by the plant. So the enzyme Rubisco catalyzes the fixation. The fixation is an important word. Of carbon dioxide by combination with a molecule of ribulose biphosphate. See, your 5-carbon compound, RUBP. RUBP is fixed by the carbon dioxide. What does it mean? It's actually just your carbon dioxide combines with this molecule of RUBP, which forms an unstable intermediate. Unstable intermediate. Since it's unstable, right? It's a 6-carbon compound. It's unstable, so it actually splits into 2 and yields 2 molecules of glycerate 3-phosphate, GP. As you can see it. See, times 2 glycerate 3-phosphate, and which is a 3-carbon compound. See, 6 divided by 2 is 3. Yeah, and from then on, it can be your GP is reduced to triose phosphate in reaction involving reduced NADP and ATP. So yeah, ATP and reduced NADP is actually used for this process. So energy is used to convert GP into TP, which we call triose phosphate. This triose phosphate is actually then used to generate your glucose, your amino acids, and your lipids. And then, since this entire thing is a cycle, right? Your trials so RUBP is regenerated from TP, from triose phosphate, in reactions that use ATP. So, in actuality, your only one out of 6 of your triose phosphate is actually used to generate your glucose, your amino acids, and your lipids. And that 5 out of 6 triose phosphates are actually used to regenerate your RUBP. Some amino acids actually are directly converted from your glycerate triphosphate. But yeah, mainly the your kind of like your glucose, amino acids, and your lipids do come from triose phosphate. And yeah, that is your light independent stage, which is the Calvin cycle. So, limiting factors of photosynthesis. What what types of limiting factors have you learned on photosynthesis?
We've already learned about light intensity, light wavelength, CO2 concentration, temperature, and supply of water.
Yeah, exactly. Spot on. Everything. Your five main factors. So yeah, as you said, what is a limiting factor? The requirement for a process to take place that is in the shortest supply. An increase in this factor will allow the process to take place more rapidly. So yeah, as you said, the five main factors: your light intensity, your light wavelength, your carbon dioxide concentration, temperature, and your supply of water. But supply of water usually affects other processes in the plant before it affects photosynthesis. And yeah, let's take a deeper dive into this. Effects of light intensity. They're very simple. So the rate of photosynthesis, as you can see here in this graph, increases as the light intensity increases, generally, like from this point to this point here, to this point actually, from this point to this point here. Why is that? Greater light intensity means more energy supplied, right? As we said previously in your light dependent stage of photosynthesis. So light dependent stages occurs faster and more ATP and reduced NADP generated to passed on to the Calvin cycle. But then, if you notice here, it actually plateaus. Why is that? What is a common thing about limiting factors is that once it is in excess, once it is no longer a limiting factor, another factor becomes the limiting factor, whether that is carbon dioxide concentration or water or anything that contributed to this. So yeah. So in general, effects of light intensity, very simple. You've learned before. The rate of photosynthesis increases as light intensity increases. So let's move on to effects of temperature. So temperature has no significant effect on light-dependent reactions because they are driven by energy from light and not the kinetic energy from the reacting molecules. But your Kelvin cycle is affected by temperature because there are more of the normal enzyme controlled reactions. So what happens at high light intensity? The rate of photosynthesis increases as the temperature is increased. Yeah, you see here, the temperature increase and your photosynthesis also steadily increases with, but in requirement of high light intensity. As we said before, uh, any factor can become the limiting factor once it is actually in short supply. But exactly. At high light intensity, the rate of photosynthesis increases as the temperature is increased. However, in low light intensity, the increasing temperature has little effect on the rate of photosynthesis. But so, yeah. However, one thing to note about temperature is that once it reaches a very certain point, usually around like 37 degrees Celsius, for example, our human body, your enzymes, the temperature is actually too high. So what happens when the temperature is too high with the enzymes is, as you learned before, the enzymes become denatured. So the rate of photosynthesis actually decreases because it's too hot. Your enzymes start denaturing, and that and as temperature increases, there are less and less of these enzymes to actually take for reactions to actually take place. So the rate of reaction decreases. So there's actually an optimal temperature in a so-called optimal temperature in which the rate of reaction occurs the fastest, as you've learned in your AS biology when you learned about your enzymes and your thing like Keith Meissner's constant. But yeah, essentially it is kind of just like common logic that you've learned before. So effects of carbon dioxide concentration. So let's take a look at this graph and compare experiment 3 and experiment 1. Why is that? Well, they occur at the same temperature, 25 degrees Celsius, and but at different concentrations of carbon dioxide. So at the start, you can see here, at the start, when light intensity is the limiting factor, see, your light intensity is in your y-axis, both increase in their rate of photosynthesis when light intensity increases. However, as you can see here, graph of experiment 1 reaches the plateau faster than graph of experiment 3. See, at a lower light intensity, your experiment 1 tree has become plateaued in the rate of photosynthesis, whereas your experiment 3 graph still requires a lot more light intensity before it starts to plateau. Why? This is because your carbon dioxide concentration of experiment 1 becomes the limiting factor at a lower light intensity compared to experiment 3, because carbon dioxide is in much less supply. Yeah, there is less carbon dioxide available in supply. So yeah, essentially that's this entire photosynthesis chapter is very short, very quick, and it's kind of similar to respiration in many aspects. And it's probably it actually is the shortest topic, in my opinion, of A2 biology. So let's move on to your workout sessions. So here's a question. Describe the process of cyclic photophosphorylation and the structure of photosystem involved. It's a 9-marker. It's a very long question, but you don't really see this see 9-marker questions now in A2 biology with the new syllabus, because they have cut it out from your past papers. But it's a very good source of its mark schemes are a very good source to make notes from, because they explain the entire process pretty much. And yeah, so take some time, pause the video to answer this question, and I'll explain it later on with the mark scheme. So yeah, let's move on to the mark scheme. So as I said before, your cyclic photophosphorylation includes only your photosystem 1, and what happens is your light energy is absorbed by your accessory pigments, and the light energy is transferred to your chlorophyll A in your reaction center, which results in your electrons in your chlorophyll A being excited and your electron emitted from chlorophyll A. And your and the electron goes through a chain of electron carriers, which is your electron transport chain, resulting in ATP synthesis from what do we call chemiosmosis. And your electron returns to photosystem 1, back to your reaction center. And photosystems, let's talk about the structure of photosystems. They are arranged in light harvesting clusters. Yeah, we call your photosystems light harvesting clusters, which and your primary pigment, another word for chlorophyll A, we call chlorophyll A the primary pigment, uh, uh, resides in your reaction center. And yeah, accessory pigments, uh, your photosystems have these accessory pigments, chlorophyll B, carotenoid, uh, stuff like that, and your xanthophyll also. Uh, and yeah, this is quite an old mark scheme, but yeah, your accessory pigments, they have your accessory pigments, chlorophyll B, carotenoids, and your xanthophylls. And photosystem is located in your thylakoid. Let's move on to another question. In flowering plants, the light-dependent reactions are carried out by photosynthetic pigments, which fall into two categories: primary pigments and accessory pigments. Outline the role played by accessory pigments in light-dependent reactions. So what do accessory pigments actually do?
So they would absorb light and pass it on to chlorophyll A, right?
Correct. Essentially, what we've learned before is, yeah, it absorbs light. And the accessory pigments actually absorb light and pass the energy to your reaction center, which where your chlorophyll A actually is, and thus it releases your electrons for photophosphorylation. Anyway, that's the end. So of photosynthesis chapter, and yeah, so
Yeah, and thank you for tuning in. We hope you understood the topic very well, and you could get hold of the notes on the ZNotes website, and our social handles are given on the screen. So you contact with us if you have any questions, leave them in the comment section, and yeah, thank you so much for joining.